Dragonet Prophecy. I can't remember. We had the Dragonet Prophecy, the Jade Mountain Prophecy, and the Lost Continent Prophecy.
Did everybody in your house read them so far? Or
Nope.
You're the
just
one.
me.
You're the one that's the big fan.
Yeah.
Very cool.
My friend across the street introduced them to me.
Okay.
Oh my gosh, these are so good. I'm not actually reading them. I listen to the audiobooks.
That's still good.
I have a CD player in my bedroom, so when I go to bed.
Yeah.
I also listen to the 39 Clues on my CD player.
Do you have to put your earbuds in or something? Everybody in your room's listening?
Nobody is sleeping in my room.
Oh.
It's just me. Katie and Joy go to bed at like.
So they come in later, so yeah, it doesn't matter. Gotcha. Okay. I was gonna say, do you let that run all night? Katie and Joy are like getting some new stories in. Yeah.
Katie likes the 39 Clues actually. She introduced it to me.
I saw those when the boys were little. I think I got one from the library for Cole once, but he didn't read it. So
I'm not one for reading big graphic novels.
Yeah.
Not graphic novels, big chapter books.
Mm-mm.
The only big one that I've ever read, but this thick, was one called Midnight on Strange Street. It is my favorite book ever in the entire world. And I gave it to Cameron, the neighbor who introduced Wings of Fire to me. And I told her, you should read this. I'm like, I can't. I'm doing this special thing at school. And I have to read like 30 book series right now. Because she has to also reread the Rings of Fire.
Oh my goodness.
So
where does she go to school?
She said, I think it was Shiloh Elementary.
Okay, yeah. Wow.
Yeah.
That's a lot of reading. My goodness.
So I finally convinced her to read it, and I lent her my book, and she's like, oh my gosh, this is so good. She got halfway through and she's like, okay, I can't keep having this. I got one from the library. I'll just read that one.
Oh, there you go.
And then like the day she finished it, we did a whole chat online about how, like about the parts of it. And I hadn't read it in a long time, so I had to like look.
You had to go back and refresh your memory. Yeah, that's fun. Hey, Louis, welcome back. Hey, Darcy. How are y'all?
Good.
How was South Carolina? Was it good?
Awesome.
Fun times. Where did you go?
Hilton Head.
Hilton Head
We were on
Island.
the beach for like 8 to 8 hours a day.
Last
Nice.
night at like 8 o'clock, my dog, my mom just let him off in the garage right next to the place where we usually let him off.
Oh my goodness.
And then there are these two crazy vicious dogs.
Oh, no.
It has a sign that said beware of dogs.
Oh no.
And then the dog runs down there, starts jumping at the window. He runs around the house trying to get out. I mean, it's funny because my dog's like not as tall as this table.
Yeah.
And then he runs up onto the street. It's like a cul-de-sac. And
Yeah.
then he goes downhill.
Yeah.
He runs all the way down onto the main road. And then he comes running back.
So your dog's okay? You just scared him?
Yeah.
Okay. That's
Mom
fine.
just called me and said the dog ran away. So I just start freaking out.
Oh no.
The cat ran away last night.
Really?
I think actually not last night, but yesterday morning we had people who are coming to replace the floors because we weren't done yet.
Oh yeah.
And they kept opening and closing the door and the cat got up.
Did you find him?
Yeah.
Okay.
We got it.
So my friend was visiting her. I told them I had a cold last week, so now I have laryngitis, but it wasn't COVID, it was just cold. But my friend went to go visit her father's lake cabin 2 weekends ago, and her neighbor called her and was like, we have a problem. Your cat's out, your front door's wide open, and one of your cats is outside. So she could find one and not the other two. She has 3 cats. And it took like all day for the neighbors to find all the cats, and nobody knows how her front door got open. It's still a mystery.
Just wind.
Well, it was locked for sure. So it's really weird. It's like, how did that happen? So they wondered maybe her landlord unlocked it, or I don't know. But I asked her if she wanted me to go over and check the house. And she said, well, my neighbor's already looked for the cat. I said, no, I mean, check the house like for your valuables and things. Your door was open. She's like, oh my gosh, you think of that. I'm like, oh my goodness. So
The cats are cute. My cats are my only valuables.
there you go. Yeah, pretty much. All right, Drew, take care of that pile, please, quickly in the next 1 minute before class. Thank you.
I couldn't zoom in last time because I was so sad. I was eating breakfast.
Oh, were you? That's okay. I think y'all, I saw somebody commented. It was probably you guys. I haven't looked yet, but yeah. Mhm.
I had my little sister wake me up this morning because I kept waking up late. and like forgetting that today is Tuesday.
Well, you've been in a time warp because you've been in you've been in a time warp with this COVID in your house forever now. So that's rough. That's really rough. Yeah. So I'm going to try not to yell too much today if I can avoid it, because this isn't good. This was what came first. And I was like, oh, allergies. And then, because this always happens with my allergies. And then it turned into like a awful cold. It was bad, but it wasn't COVID because I checked it. And then it went away and I still have this. So now I have some steroids because I've got to get back my voice back by this weekend because I need to sing. So I'm going to hand out some things to you guys, stuff to hand back mostly. And some of this you got, like this is your progress report and you got it by email, but that's your hard copy. Drew.
Oh, so Wilson.
Lily.
Is Lily gonna be joining us today?
Lily is not here. Lily's grandfather is very sick and they left town to go see them. So she'll be out at least this week, maybe next week. I'm not sure yet kind of where they're gonna land. We have a lot of family stuff to take care of. Okay, Darcy, Lewis. They were planning on going tomorrow, I think. And then her grandfather got a little sicker, so they had to like very quickly. This is your. Yep.
10 out of 10.
Ooh, 10 out of 10.
And it's for John.
Graco scoreless.
Here you go.
Since I saw
Right.
scoreless on its back.
Hey. All right. Drew, coming. Drew can't keep his stuff cleaned up, so we're gonna start.
People call that. I wrote it on like the Google Doc one, on a Google Doc once, and it auto corrected it.
Hello, Lily. If you're there, whenever you get there, because you're probably on a plane. And Sky, I don't know where Sky is. All right, so my scratchy laryngitis post-cold voice is gonna do its best today. All right, so today we're talking about DNA. Last week we talked about genetics. This week we're talking about DNA itself. And next week we're gonna talk a little more about how DNA makes the code within it come to life, how it makes it into a physical thing, like a trait. So your lesson for this week, this next one, lesson 11, is there's no reading, it's videos. It's a couple different YouTube videos and some stuff for you to fill out in your binder.
We showed my mom and my sister's video, and she just watched it.
Aren't they great?
I know.
I love them.
Wait, is this one lesson 10 or lesson?
This is 10. We're in 10.
Kitty bear.
All right, so the cell and inheritance. So we're gonna go over the stuff that you guys did with your notes and your reading, and then we're gonna add a little extra because I'm gonna give you a little history background on DNA. So crossing over. You guys probably read about crossing over, and it's a process that produces new combinations. We call them recombinations of genes. And what it does is it interchanges the corresponding segments of non-sister chromatids on homologous chromosomes. So it occurs during prophase 1 of meiosis. So do you guys remember last year what we used as kind of like an analogy for sister chromatids that go together, homologous pairs? Do you remember what we talked about? We had shoes in our image. So we had shoes and we talked about how if you have a pair of shoes, like I have these boots, and they're not identical because one's a left and one's a right, but they're a homologous pair. They go together.
What if two people had the exact same size feet and they're in the exact same shoes and they purchased it at the exact same time, they look the exact same, and they had the exact same right shoe? Then it would be a pair.
Maybe like a copy, yeah.
Like a twins, they have the same DNA.
Twins do have the same DNA, correct? Well, identical twins, not fraternal twins. All right, so what we're gonna do is this is a quick little video. It has a little graphic on what crossing over looks like. So let's watch this. Hope my volume's up right, I'm not sure.
The process of crossing over, which occurs during meiosis, is one of the most important genetic events, for it guarantees the individual characteristics of each one of us. When two homologous chromosomes lie side by side, breaks occur in the chromosomes and corresponding lengths of the chromosome are interchanged. This crossing over disrupts the original sequence of the alleles and interchanges parts of both maternal and paternal chromosomes so that the possible number of gametes with different genetic information that any individual can produce is almost incalculable. This is why a child may inherit his father's nose and his mother's eyes.
So without crossing over, we have sort of like a limited number of combinations. But crossing over is kind of a random event. It doesn't always happen. It doesn't always happen with the same loci on chromosomes. So it can be different genes that are moving around. So it doesn't always happen. And when it does happen, it's a random thing, which means that, like it said, the number and type of gametes that can be produced are almost infinite. So you can make one organism can make almost an infinite number of completely different sex cells. So all of their offspring have the potential to be completely different. All right, so
The process of
yeah. How do you mean?
But like, how is it possible for a guy, for example, to make billions upon billions of different DNA sperm cells?
Yeah, they're all different. They're all different. Because so if you start with when we do meiosis, we did a little meiosis last week, right? Where your cell divides and you end up with 4 gametes, right?
Yeah.
When you end up with 4 gametes, there is a certain number, I'm sure you could find out if you did some fun math, a certain number of possibilities you could have without crossing over, where the chromosomes all, your 46, 23 pairs sort randomly each time, right? They sort independently, independent segregation. And so those are all different. And then they can have crossing over occur, which means that they're even more different than they were to begin with. So it's almost infinite, the number of different gametes you can produce. So just to kind of keep the big picture in frame, can somebody tell me why sexual reproduction is advantageous for a species?
Advantageous.
Yeah.
Necessary?
Why advantageous as in why does it help us? How can it help a species survive as opposed to asexual reproduction?
Because asexual reproduction means you do it yourself and you're making a bunch of clones of yourself and there's no diversity.
Yeah, there's no diversity. So why is diversity advantageous? Think about it.
Because if one has a problem, all of its offspring.
Yeah, that's true. If you get a mutation or something, they all have it. What else?
Oh, like in Wonder, Olivia and her mom and dad, they all had like this mutant gene. It was just like super bad luck. It showed up in Wonder Kid.
Okay, yeah. So another reason why sexual reproduction is so advantageous is because all of the offspring are slightly varied. They're not, like you said, Drew, like clones of the parent. And so if you have a change in the environment, chances are some of those offspring are going to be better suited for surviving. And if all of your offspring are the same, there could be a change in the environment that they're not suited for, and you could lose all of your progeny. You could lose all your offspring, your whole genetic line dies out, and you're done. So that's why sexual reproduction is so advantageous, because it produces offspring that are all slightly different, and you can end up with some that are able to survive whatever changes are presented. And those changes could be anything. They could be weather, climate, they could be about food, and it could be even illnesses like we're dealing with right now, right? We've had a pandemic. Some people are going to be better suited to survive than others. Like if we didn't have the modern medicine we're using and the, all the different treatments and the vaccines and stuff that we're using, if we didn't have any of that, there would be some humans that are just genetically better suited to survive than others. And those would be the ones that carried on, and the others would die out. No.
I have a pen pal that her sister has Down syndrome. So she's still in full quarantine.
Oh yeah.
Because of her sister.
And we're going to talk about Down syndrome in a couple of weeks when we start talking about problems with chromosomes and things that can happen. So transcription. So we have two main things that happen. Hey, come on. We have two main things that happen with DNA and with RNA. They're transcription and translation. Colson, can you scooch over a little, buddy? There. Anthony's skinny, but I don't know if he's skinny enough to want to sit right on top of him. Yeah, I like that. Did you change it? I like it. It's cool. It's what? Oh, yeah. So you had to untwist it. Did it take you a while? Yeah, it looks good. See, he comes in looking good. You need a haircut. I've been telling you for days.
I love my hair.
Yeah, yeah. All right. So what we just talked about, Anthony, was we talked about crossing over, which you guys had read about, and about how one section of a chromosome can flip over and join another section of a chromosome. And I already apologized for my voice. I've got laryngitis because I had a cold last weekend, but it's gone now. It's just leftover laryngitis. All right. So transcription and translation. So let's talk about transcription for a minute. So if you remember from your reading, transcription is the process by which the information in a stranded DNA is copied into a new molecule of messenger RNA, or what we call mRNA. So DNA safely and stably stores genetic material in cells as a reference or a template. So it's kind of like a dictionary or an encyclopedia. All the information is in it. What?
Also complicated.
Oh
Ew,
my gosh.
I'll get it. Here, I'll grab it. I hate palmetto bugs. They're so grody. Here, I got a tissue. Grab- Oops. They're so huge too, because I have all these big. Yep, yep.
Ah.
We just find these random giant cockroaches in our house.
When I
It's
went
so
to the
gross.
library, the other day, there was a dead cockroach in the
Got him.
bathroom floor.
Got him.
It sounds even worse than the gas station.
Yeah, you know, the first time I moved here, I had never seen them before. And there was one that showed up in my house and I freaked out. My uncle was an exterminator. And I called him and I was like, what is this thing that's in my house? And he had to tell me what it was. I had never seen it before. All I knew is it looked bad.
When I moved here, I stepped on a huge amount of fire ants,
Oh,
and my mom didn't know what they were.
That's
Somebody-
bad.
And they're just like crawling all over my
Somebody
house.
dared my little sister once to sit in an ant pile, and she did it.
Who would do that to Ellie?
How much money? How much money?
That's not
20
nice.
bucks. 20 bucks.
Hey, that's not bad. That's not, I did 20 bucks. That's not
If
bad.
it's
I
20 bucks, it better be.
The
more than
crazy
that.
thing about fire ants, right? So ants are social insects, and they'll do this lovely thing where they'll crawl up on your body
Oh.
and then they'll wait until one of them says go, and then they'll all bite you at the same time.
Wow,
And you don't know they're there until, because I did that once. I was standing in one and I was outside and I had on like a short top and pants because I was doing some garden work. They were all the way up to here on me before they started biting. It was not fun. So anyway, let's get back to this lesson. So your DNA is like an encyclopedia. All the information's in it, but it can't do anything with that information without the help of RNA. So it just stores it all. Hey, Sky, come on in. So your DNA is storing all your information, but RNA is able to make a copy from it that can actually be used. So this is the process of transcription. Transcribing, like if you're writing, transcribing. Do you know what a scribe is? Anybody ever heard of like a scribe? Sometimes you hear the word scribe and talks about like historical figures, or sometimes you hear the word scribe in Bible readings too. Like there are scribes. Scribes are people that translate. Like they'll take something written in Greek and they'll translate it into Latin, right? So transcription is exactly that. It's the process of translating what's in the DNA into an RNA molecule that can actually do something with it. So meanwhile, messenger RNA is comparable to a copy from a reference book because it carries the same information as DNA, but is not used for long-term storage. It can freely exit the nucleus. So although messenger RNA contains the same information, it is not an identical copy. That's because it has some different nucleotide bases. Because its sequence is complementary to DNA templates. So look right here. So your DNA has 4 letters, right? We talked about our 4 bases. What are they? What are the four letters in your DNA bases? T,
A.
T, G, A, and C, right? A and T go together.
Right there at A, it says og.
Uh-huh.
If you added an E,
GGIE, yeah, be your brother's name. So yeah, so your A goes with T and your C goes with G and vice versa. So if you look right here, you're looking right here, you see an A goes with a T, T goes with A, G goes with C, C goes with G. You see that? All right, next week we're doing a lab where you're going to do some translating. It's going to be fun. But RNA is different. Remember, these are adenine, thymine, guanine, and cytosine. There is no thymine in RNA. There's a different nucleotide base called uracil, and that's this U. The U doesn't exist in DNA, only in RNA, and it replaces what? Can you tell? What's missing? Can you tell what it replaces?
C.
Look right here. What's it paired with?
A.
It's paired with A, so what does that mean it replaces?
C.
Nope, T. There's no thymine. So uracil replaces thymine in RNA. So that's transcription, okay? Transcription is this process right here where we're gonna take the DNA and we're gonna turn the code into an RNA code. So it's just like you went to the library and there was a book in Spanish and you're like, oh, all the information I need is in this book, but I can't take the book out of the library. Your nucleus is like a library. You can't take the DNA out of the nucleus. You can't take the book out of the library, but you need to make a copy of what's in it because you need that information. So you sit down with your pencil and your paper and you take what's written there in Spanish and you write it in English. Like maybe it's a little story and it says, Juan went to the market to buy some apples. He paid 5 pesos. And you write that in English because it said it in Spanish in the book. You write it in English and now you can take it out of the library. You can take it out of the nucleus. That's what messenger RNA does, okay? That would be you being messenger RNA. So now translation, that's transcription. Now translation is different. Translation is the process of translating the sequence of mRNA into amino acids during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes. All right, so this works. If you remember last year, we did this a little bit. So it works in sets of 3, and that's called a codon. So 3 nucleotide bases is a codon. And what happens is you'll get an exon and an intron, and that basically tells it where to stop coding and start coding. It's like a little sign in the writing that it has that says start here. And then it will take that information, and it would be almost like you, if you were to take that story and then act it out, make it into something you could actually see that you'd previously taken from Spanish to English, and now you're acting it out, it would be that. So what happens is your RNA has these, see there's 3 here, UAC, AAA, GCU, and it takes every one of those is a different amino acid. And amino acids are the parts that make up a protein. So when you have a code like this, it'll start to build a string of amino acids that will build a protein. And that's how you get your eye color, your height, your hair color, your whether or not you like certain foods that can be genetic. There's some tastes that are genetic. There are some, like some people can taste certain chemicals that other people can't. Like who can do this? Who can roll their tongue?
You can flip the tongue upside down.
Oh, I can't do that. Can anybody do that clover leaf thing? Some people can make their tongue look like a clover leaf.
I can do that.
I think I can do that.
Can you do that? Almost, yeah. That can do this,
Since first grade.
but I can't do that other thing. It's weird. And then like, look at your earlobe. Is your earlobe, mine's attached. Some people's hangs down, not attached. Yours is free. Mine's attached. Yeah. Yep. So that's a genetic thing too. All that comes from a code of proteins. So you can have mutations, and we're going to talk about this more in a couple weeks. But mutations occur when DNA in a gene is damaged in such a way as to alter the genetic message carried by that gene. And sometimes it's not even damaged, it's just a mistake in coding. Have you ever copied like a really long number and you messed up a number? That happens, right? Yeah, that happens. Or say you had, say somebody gave you like a locker, a combination for a lock, and it was like 30 numbers long, and a drop of water got on the middle of the ink, and you can't quite read that part in the middle. That would be like damage to DNA. And DNA can get damaged in lots of ways. When you hear about things that cause cancer, like a lot of those things damage DNA. Okay, so a mutation occurs when it's damaged in such a way as to alter the genetic message carried by the gene. A mutagen, that's what you call a substance that can bring about a permanent alteration to the physical composition of DNA's gene, such that the genetic message is changed. So if you look right here, this is changed. This shouldn't be here. And this is called a point mutation. It means one thing is wrong. One tiny nucleotide is wrong.
Well,
Now, here's the interesting thing. You are each full of billions of cells, right? And those billions of cells all have DNA in the nucleus. And then you get, especially your somatic cells, your body cells, okay? And they all have DNA in the nucleus. And then your DNA is made up of billions of these base pairs, right? You have many mistakes in your DNA, many mistakes in your DNA. And most of them don't cause any problems that you can see. But some of them cause huge problems. It just depends on what gets mutated, where it's mutated, and how important it is to the process. So some mutations are just there and you don't even know they're there. A lot of mutations are there. So this right here, I'm going to show you a video here, and this is going to show you translation.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
How long does that all take?
It happens constantly, like
Seconds.
always. Yeah, it's always going on.
Now.
Yep.
Now.
No.
I know it's
No.
not.
No. Yep. And so when you think about it, like think about how when you a good example is think about when you cut yourself, like you cut your hand. Okay, you can watch your skin heal back. right? That's all building proteins. You grow in height, that's building proteins. But you also, it's really crazy to consider. Do you remember how many amino acids they said there were in the video? So it's 20 amino acids in total. And your body can make, with those 20 amino acids, nearly every protein you need. There are some that you take in with diet, but it builds these proteins and they're all different. And it has to do with the order in which the amino acids are assembled. You can assemble them in a certain order and it produces one protein. You assemble them in a different order, it produces a different protein. And that's how you get, because it's proteins that make your veins, your cell walls, your hair color, your all of the neurological functioning is all structure is all protein. So everything is basically made of protein. So it's all comes down to these 20 amino acids that are coded for by 3 little letters in your DNA strand.
How much protein would it take for me to grow 5 inches in 1 year? Because I had done that.
I don't know, but you probably ate a lot. You probably ate a lot. You had a lot of energy, I'm sure. Yeah, Drew's working on it. So this part wasn't in your book, but I want to give you a little bit of background so you can see how we got to where we are. So you can understand how we know what DNA looks like and how we know how it functions. So we'll go back to 1928. So a little over 100 years ago. That's not that long.
Physics and pictures.
Yep. So this is not that long. There are people alive now who were born around 1928.
My great grandma.
Right? Yeah.
She lives with us.
She would have been like what, 1924 or something? No, 1930, 1930 something, 1932, 33. Yeah. All right. So
she was a teenager during World War II.
Yep. So this guy, Frederick Griffith, this is Frederick Griffith. He looks very stern, doesn't he? So in 1928, what he did is he worked with mice and he worked with a bacteria that caused pneumonia. And he had 2 different kinds of bacteria, one that caused pneumonia, one that didn't. And then he would kill off the, he would heat kill them. Yeah, no, not the mice, the bacteria. He would heat, well, the mouse did, yeah, a lot of the mice died. But anyway, he heat killed them and then he would combine the strains and he did all this mixing and stuff in his lab. But what he concluded, the big important thing is that genetic information could get transferred from one type of bacteria to another type of bacteria. And he called this transformation when one type of bacteria, which is harmless, is suddenly permanently changed into one that causes a disease. And he decided that the factor causing that transforming must be, he's going to call it a gene.
I remember saying, my mom reminded me when I was about maybe 8. Anyway, since a lot of people were getting cancer and stuff.
Yeah.
I was like, well, maybe they wouldn't get cancer if they didn't have cancer cells in their body, but their body won't function right without cancer cells. And then I thought, what if people reduce the cancer cells in size?
That's what radiation does. It shrinks them. Yep. So basically all these mice that weren't supposed to get pneumonia and die got pneumonia and died. And he's like, oh, there's something doing that. So the next guy that came along is 1944. So that's a good jump. Not quite 20 years, but a little further down the line. And we get this guy, Oswald Avery. Okay, Oswald Avery worked with 2 other guys, Colin MacLeod and Macklin McCarty. So these are some good Scotsmen here.
Colin doesn't look very happy.
Yeah. Yeah, Colin looks like he's being distracted. Colin MacLeod, Macklin McCarty, and Oswald Avery. Ooh, there's a typo.
Avery looks a lot happier.
Now fix that. Yeah.
You have a
There you
connected
go.
keyboard.
Yep, clicky clacky. So what they did is they basically did a process of elimination. So Griffith has said something's causing a change and it's called a gene. So they wanted to know what the gene was. If the gene was a protein, if it was a lipid, if it was a polysaccharide, or if it was a nucleic acid, which they knew existed in cells because they'd been able to analyze the makeup of cells, but they don't know what's doing what. So they discovered that DNA is what stores and transmits information from one generation to the next. And they decided that the DNA is what makes the gene that's responsible for the transformation that Griffith saw. And they did this basically by taking cells and mixing them with all these other things, proteases that chewed up the proteins, lipid chewers, polysaccharide chewers, basically things that eliminated one of these substances from the mix till they could find out that it was a DNA. Then you get these two. So this is Hershey Chase. So this is Martha Chase and Alfred Hershey, right? All right. So what they studied were bacteriophages. Now, bacteriophage is this funky looking thing right here.
Looks like a robot.
They do look like robots. They're really cool looking. So a bacteriophage is basically a little strand of DNA coated in protein. So it's a virus that infects bacteria. It's not a virus that infects you. It's a virus that infects bacteria cells, basically. But they're itty bitty bitty. And they'll land with those little legs like a lunar lander on a bacterial cell, and they inject their DNA into the cell, and that makes the cell reproduce its DNA and spit out more bacteriophages.
Like the cookie robots are Despicable Me.
Yeah, like that.
Cookie robots.
Yeah, cookie robots. So they studied bacteriophage composed of a DNA core and a protein coat, and they wanted to determine what part entered the bacterial cell. Was it the DNA or the protein? So they used radioactive markers and they labeled the DNA and the proteins. And then they were able to tell when they had allowed them to infect the bacteria, they were able to tell whether it was the DNA that went inside the bacteria or the protein that went inside the bacteria.
I wonder if radioactive crayons draw like neon colors.
Oh. Some do use neon. Use like, wait, is it called neon? Isn't that?
I don't know. What is it again? Say it again.
What was that material that radium?
Oh, radium.
If you use radium, that will fill in that colors.
Oh, right. Okay. So radium. Yes. So a good historical example of not understanding things well yet is radium. So radium, radium is radioactive. It's not good to be around, but it makes, if you put radium in paint, it will glow in the dark. And back around the turn of the century, it was very popular to have radium painted on a watch face, on the numbers. So if you were in the dark, it would glow. And people, oh, it's so cool. So there were, in New York, especially, was one of the big centers for this. And there were girls, and these are girls that are like probably 12 to 20 years old that worked in these factories. And they were hired for their skill in doing very, very fine work. Like they would paint tiny, tiny numbers with a tiny, tiny brush. And they were told the way to get the best point on the brush was to put it between their lips and pull it tight.
Oh no.
And they did that with radium paint day in and day out for years. And many of them ended up dying, but not just dying, like their whole jaw would dissolve. All of their jaw would dissolve, their bones.
And all the people who
That's a
did
Parts of their
Picture
skull
in
would
my
fall
brain.
apart while they're still alive. Like it was awful.
I'm gonna have
Great
nightmares.
picture in my brain.
Yeah. And it was terrible. And it was the kind of thing that, like the company ignored for a really, really long time until they were kind of forced to acknowledge that it was happening.
Great job, government.
Mhm.
Alcohol
Well, it was private
testers-
company. Yeah.
Alcohol testers have to put both of they have to put it in their mouth. They have to switch it around.
And spit it out.
They can't swallow it because they'll become drunk.
Yep.
If they do.
So now we have Erwin Shargraf. Okay. So this guy basically, he came up with an idea that gave really, really important information. So what he did is he isolated the different nucleotides in DNA and found out that there's always equality in quantity, so the same amount, between A and T and G and C. So think about it. If you have 4 things in a pot, and when you pull them out, you find there's always the same amount of A as there is of T, and the same amount of G as there is of C. You've learned that those go together, right? Those are always the same. So that's really, really important, because without that information, This next bit of stuff wouldn't have made sense. And this was 1949. So most of you who have grandparents, your grandparents were alive at this point, for most of you. Like my dad's 1942, my mom's 1946, okay? So now we're gonna talk about this lady, because this lady suffered a huge injustice in the scientific community. This is Rosalind Franklin. And this thing right here at the bottom, this is photo 51. My dress is smack covered in photo 51, okay? It's one of my favorite images. So she had a lab, and in her lab, this guy, Raymond Gosselin, who was one of her students, okay? He was like a postdoctoral student working in her lab assistant. And it was following all of her instructions and taking all these pictures. She was using X-ray diffraction, and he took this picture. He's the one that actually snapped it, but it's all her process. You girls paying attention? So the diffraction pattern here showed something that nobody had figured out. What this is it's looking at DNA from the top down, like you're looking through a tube, straight down. And you see this little X here. And then you see that in the distance, it kind of repeats, but you don't see it as well because it's far away. But you see this X. And that's how we knew that it is a double helix. There was nobody had thought that yet. Nobody had figured that out at all. So when she got this picture, she saw that these strands were anti-parallel, and the outside of the DNA chain is the backbone. That's your deoxyribose and your phosphate groups. And then your base pairs were like a ladder, but it was twisted. And that's what people couldn't figure out. And this was in, check the date, 1952. Okay, now let me tell you something about Rosalind Franklin. First off, in the 1950s, for a woman to be in science was rough. It was rough. It was definitely a boys' club. Okay, definitely a boys' club. And I'm not just saying that to say that. And Rosalind Franklin was not a girly girl. She was not. She didn't do the things that society thought women should be doing necessarily. In fact, she balked in the face of it all and was like, you know, she didn't even didn't even apologize. She was totally unapologetic for being what many people considered brash and abrasive. She was not soft-spoken. She didn't stay in her place. She said what she wanted to say, and she didn't say it necessarily nicely. She just said it. And the men that she worked with found her very, very off-putting. They didn't like her at all. A lot of people didn't like her. She wasn't a friendly, warm, cuddly person at all, to be honest. So then what happens is this dude comes along. Now note the year, 1953. Right? Yeah, he's a spy. Maurice Wilkins is a spy. And here's what Maurice Wilkins did. Maurice Wilkins took Rosalind Franklin's research to Watson and Crick, who had been spending all this time trying to build a model of DNA, and they just couldn't figure it out. They could not make it work until Maurice Wilkins showed up and said, look what I have. Gave it to them, and they went, oh, a double helix, and they built that. And they got the Nobel Prize, and they got put in every textbook, and they got all the credit for discovering the structure of DNA. And until recently, like really recently, society didn't acknowledge Rosalind Franklin's contribution that much. In fact, when the research was published, they published it all in a book that year of like research symposium research that went out that year. Theirs was here and hers was underneath. So it looked like hers came after theirs. Now, at this point, she had moved and left her lab, so she didn't have much recourse. And then she died very shortly after. So she didn't get the Nobel Prize because it's not awarded posthumously. It's not awarded after you die. So here's a video about Rosalind Franklin, because she really, really should get more credit than she does. Nowadays, we're kind of making up for it. We're making up for it a little bit these days. Yeah, these are great.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported.
Well, they could have given her credit. They actually chose not to.
Or that she was a plain dressing, belligerent scientist. which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind Elsie Franklin, the unsung mother of the double helix.
She was actually very, very cool. Very cool, but didn't make friends. She wasn't a nice, friendly, warm, cuddly person.
Well, I mean, still though, it sounded like she was a jerk, like kind of mean.
Maybe a little mean, rough around the edges, but to get where she got, she didn't get there by being nice and friendly and warm. If you were nice and friendly and warm and did all the things you were supposed to do, you would, go shut up and do your needlepoint. And she didn't do that. So, yes.
Did you get my picture that I sent you of my homework?
I think so. I'm not sure. I'll check though. All right. So this, if you want to check your homework, hand it in if you have it. But if you wanted to look over it, this is what we've got. And some of you already handed it in because some of you had it done early last week, and that's fine. What?
I think I left it at home.
That's okay. Just send it to me when you get home.
Okay.
As long as I get it by the end of the day, it isn't like.
I did?
Yeah, because we were working with it in our binder.
So we talked about it last week, but here it is again. And last week we started a little bit with doing this little thing here where we're gonna design some dragons, use some DNA, and we're gonna keep working on that. Also, something we're gonna do, something we're gonna spend some time on when we have a little gap. Today we're going straight to lab, so we don't have much of a gap. But this is a 3D model of DNA right here. with the paper. And basically, when you stick these together correctly, they have little tabs. And when you stick these together correctly, it starts to build a DNA model. So what we'll do is, as we get time, we don't have much time today, but as we get time, we'll build with these and we'll see what we come up with. But we should, excellent. There's a folder up there. You can put it in the folder. So today for lab, we're going to start in this room. So if you guys want to just kind of get lab out and get ready, we'll start from here for lab. I'll let Lily go.
Yes, I'm done with that.
Yay.
Anyway, we can
Okay. All right, Lily. Catch you later.
These are my favorite. She introduced me to this series.